Awesome.
Yeah.
My name is Han. I'm currently a PhD student with Francis, Tim, and Mike. And I'm just going to be talking about what I'm currently working on. So there will be some preliminary results and the gender control summary responses to low humidity. Before I start, it's not changing. Awesome. I want to acknowledge the Tasmanian and Aboriginal Palawa people on the land on which I work. I want to pay my respects to their ancestors and the descendants. And the language, the Palawa County, the cultural and spiritual connections. And I also want to pay my respects to the traditional and original owners of the land, the Mowinia people, and to pay respects to those who have passed before us. So just a very brief introductions about the fact that plants are important in our natural and control system. I don't think I need to convince anyone about that. The fact that over the past few decades, we are seeing a dramatic increase in extreme heat events, with the air getting hotter and drier, making plants more susceptible to dehydration stresses. On top of that, there are growing demands of growing populations, and we need to at least double our plant productivity. However, even on a daily basis, low humidity, the air humidity can change really suddenly, and low humidity is a big stress for plants. However, we don't really understand the importance or like the fundamental aspects of like the plant biology that underlying the productivity and responses in challenging low humidity conditions. So I'm trying to address one of those knowledge gaps. So looking at the mechanisms underpinning the rapid response. Most plant tissues are covered in a thick layer of cuticles, which are great with limiting water loss, but not so much for preventing CO2, for getting CO2. Plants need CO2 for photosynthesis, so they can acquire it through adjustable pores called stomata. However, because the air inside stomata is a lot more humid than the air outside, so water also evaporate through those pores. In dehydration stresses like low humidity, there's an increase in transpiration through stomata. To combat this, most plants can increase the biosynthesis of a stress hormone called ABA, and ABA triggers stomatal closure to limit water loss. So these are just the major lab plant groups. I'm just going to have a quick introductions of what we know. It's basically, we know that the modern day algae, green algae, show that ABA can be used for dehydration tolerance, suggesting that the ABA for dehydration is conserved across land plants. However, so far only seed plants, so gymnosperm and angiosperms, have been shown to use endogenous, meaning self-made ABA for stomatal closure, and only angiosperm shown to have ABA rapid ABA biosynthesis for rapid response. So what do we know about the ABA biosynthesis? We know a lot from various different tests before, especially in Arabidopsis. So we know that it's multi-step pathways, and that the precursor, so precursors before synthesis are plentiful, suggesting that the rate limiting step is catalyzed by this enzyme called NCED. NCED gene expressions have been shown to correlate really well with ABA levels. So in my lab, Frances have shown that when she was investigated in the speed of response on Arabidopsis, actually looks at NCED, ABA, and stomatal closure, and what we see is that the NCED gene expression actually induced within 5 minutes of stress, ABA within 5 to 10 minutes, and stomatal closure is within 20 minutes of stress perceptions. So, but then we don't really actually know how it's being regulated. So we have an idea of what kind of components that might be in the pathway. So normally we have the cell membrane touching the cell wall, like in this case. But in dehydration stresses like low humidity, the cell will lose water, and the cell wall will kind of cell membrane kind of shrinks, so pull away from the cell wall, changing the cell wall cell membrane relationships. We thought there might be some kind of sensors to sense change at molecular level in the cell wall cell membrane relationships. There might be some signaling molecules in the middle to amplify and transmit the signal to the nucleus, where it activated the transcription factors to change the gene level of NCED, which increase NCED protein level, and then upregulated ABA biosynthesis. But we don't really exactly know the mechanisms for inductions again. So that's a big M. I'm wanting to know the genetic control of somatic responses. And one of those sub-aims, which I'm going to be talking about today, is examining the rapid responses to low humidity through transcriptomics. And the reason I want to do that, I want to see all the different gene expression changing at different time points across the first 20 minutes, what pathways they're in. And then I can possibly use that information to identify the different ways of gene expressions and maybe identify some candidate genes for these pathways that we just saw. And most importantly, I want to get insights into the earlier protein interactions, like for example, what transcription factors are being activated and binding to to cause the gene expressions. Especially around the NCED upregulations. To do that, I basically collected for 8 different time points spreading across 20 minutes, as shown here, to characterize the RNA responses. So after collections, I have a lot of genes that are changing differently in response to stress. And I want to have a way to analyze them, because that's the whole point of collecting so many. There's a new package that are currently being developed by Luke and Co. If you are interested, feel free to scan the QR code. That can help to understand how and when things are changing. So taking this genes example, you can obviously see there's an increase, but like when. So CPAM can tell you when the changes, in this case is a 2.5 mark, and the change is basically continuous, so there's a continuous increase. So personally using CPAM, I can see all the various different response shapes, whether it's in different time points, whether it's increase or decrease, and what type of increase and decrease as well. So whether it's a continuous change or is it plateau at some point, or it could be just a transient increase and decrease.
Appreciate it. Thank you.
So all this will just help me to identify and like understand different biological processes and different pathways at different time points. This is just again, this is a experimental in progress, so I'm still analyzing all the exciting stuff following. So keep in touch. So I obviously have to look at my NCED genes, which is again the rate limiting step of ABA biosynthesis, and I saw that it's being induced at within 5 minutes of stress perceptions, suggesting again this is in line with what we've seen before in our group, and that things are getting upregulated really quickly. For an overview, I use gene ontology, which also known as coterms. It's just like short descriptions of the gene groups and tell me generally what kind of biological process, molecular functions, or cellular components that each genes are involved in. So as you can see, there are three main categories, and let's start with the biological process. We see most of them in stress response, which is absolutely what we are expecting. We also see that, more interestingly, signal transduction, so this one in here, is being enriched at about 5 minutes. So around the time point that ABA biosynthesis NCED is kicking in. So saying that it's like start quite early as well. When we move to molecular functions, I found many genes that are important for transcription regulatory, so suggesting that things are being regulated as soon as stress are being upregulated. Stress are being perceived. I also seen some calcium binding sites, calcium binding proteins genes popping up. And if you haven't known yet, calcium is actually really important for stress transition, and you usually see like calcium waves when there is any type of stress. So suggesting that there are some signaling kicking off as soon as NCED it is starting to be upregulated. Cellular component, there are some plasma membrane and cell periphery enrichment. So there might be some genes that either located at the membrane to sense the different in the relationships. So that might be the reason for the increase in the enrichment. And so it would be really interesting to kind of tease out all of these. The dynamic changes in the transcriptions and GOTAM suggested that there are a lot of things are going on, and I want to know about the transcription as stated, what the transcription factors that are active in these 20 minutes. So I look at the binding sites of it, of all the differentially expressed genes at different time points. If you don't know, transcription factors, again, bind with the gene and upregulated the genes. And it binds to a specific region called transcription factors binding sites. So these are just the common transcription factors binding sites family that I can see, because in the family, the transcription factor might have really similar binding sites. And these are the most common one, and you can see that it's actually getting more and more active towards the end as we have more and more transcription factor binding sites enriched. So yeah, there are a lot of different dynamic going on, and again, need further detangling. One way that I'm currently detangling is that I'm using the transcription factors binding sites informations to identify what transcription factors are involved. And in this case, these are the ones that are binding with my favorite genes, NCED3. Using bar, so if you are interested in using it, feel free to scan the QR code again. So it can tell me whether it has been either experimentally looked at, or it's just theoretically interactions. The bar can tell me what they are interacting with. So the circled one are what my transcription factors that I identify that binds with NCED. And you can see that there's a lot of different interactions going on here. For example, some of them even show that they can upregulate themselves with the little circle and then going back here in these two. Some of them might be interacting with each other. So that would be interesting to know, to investigate why, or look at whether there are any data, any published data about this. Or some of them might just be interacting with the same genes. So maybe these genes will be important in later time points, or in later response down the stream. It's just to note that these are just protein interaction, so it's not going to be able to tell me much about protein DNA interaction or protein RNA interactions. So this is where I would like to add for more advice, because bar is really great, but there's a limit on how much genes I can input in at the same time. So if anyone knows any other programs or any other place, it would be great. So please let me know. Oh, one more thing for bar, it can also tell you where the protein interaction is happening. So most of them is happening in the nucleus, but some of them you can see that they might move to plasma membrane or plastids for that. Anyway, preliminary conclusions, just moving everyone around, that CPAM is an excellent tool for understanding the different gene expressions, and there are so many interesting enrichment terms and transcription factor balance sites that come up. And currently in progress, I want to know all the interactions, protein interactions that either taking place before or during that 20 minutes. So yeah, well, thank you for listening. I want to end early so that we have plenty of time to discuss and talk about possible ways, possible interactome that I can use. Thank you for the funding bodies, and obviously biggest thank you to my supervisors for helping me with this.